First of all, I'd like to thank the organizing committee for giving me the opportunity to present my work today. I'd like to present my study titles, Evaluation of the Effect of Integer Assignment in RNA Inverse Folding Problem using Factorization Machine with Annealing. I'm Shuta Kikuchi, a project assistant professor from Keio University, and this work was conducted in collaboration with Professor Shuta. This is an overview of this study. As part of our study on quantum computing for biology, we have evaluated the RNA inverse folding problem using an Ising machine-based approach. To address this problem, we employed the factorization machine with annealing FMA, a black box optimization methods. We specifically investigated the effect of assignment and ordered the categorical variables to integer variables in FMA because bases of RNA, which are categorical variables without inherent order, must be represented as binary variables. The effect of integer variables with integer assignment on optimization performance was evaluated using 27 base size target range from the ETNA platform. When using domain order encoding, it was found that certain bases were generated depending on the assignment between bases and integers. So first, I'd like to describe about quantum for biology. Quantum computing is expected to be applied in various fields of biology and life sciences, such as clinical trial optimization, biomolecular network analysis, biomarker discovery, and cell-centric therapeutics. And the Q4 Bio supported challenge problem launched by Wellcome Leap is a pioneering international grant initiative designed to accelerate the application of quantum computing to biology and life science. In addition, several collaborations between quantum hardware or software companies and pharmaceutical or healthcare institutions, such as DF Systems and JT, Japan Tobacco Pharma, or INQA and AstraZeneca, the IBM and Cleveland Clinic, has recently have reported. Thus, there is growing interest in applying quantum computing to biology and life sciences. So, we challenge quantum for biology. As part of our study on quantum computing for biology, we focus on the RNA inverse folding problem. I will explain this problem in detail. First, as you know, RNA has 4 bases: guanine, cytosine, adenine, and uracil. These bases are connected by a phosphodietyl bond to form an RNA sequence. In biological systems, 37-degree pH 7, RNA sequences tend to adapt the most thermodynamically stable structure under equivalent conditions. GC pair and AU pair and GU pairs are known as typical base pair in RNA. These base pairs are held together by hydrogen bonds. The base pairing occurred because it provided us some dynamically more stable state. The formation of hydrogen bonds between complementary bases, along with base stacking interactions, lowered the Gibbs free energy of the system. Therefore, the RNA sequence adopted such secondary structure, where some regions from base pairs while other remains unpaired. The RNA secondary structure is very important because the RNA function and molecular interactions are related to the secondary structure, like this. So the RNA inverse folding problem is to design the nucleotide RNA sequence that folds into the desired target secondary secondary structure. The well-known protein foldings aim to determine the optimal secondary structure, secondary or tertiary structure that protein adapt based on its amino acid sequence. But the RNA inverse folding problem is an inverse problem in which we first prepare the target secondary structure and then design an optimal sequence. This is a difference between the protein folding and RNA inverse folding problem. The RNA in the inverse folding problem became increasingly difficult to solve. The number of bases and the structure complexity increase. This table shows the base size NT. NT means nucleotide. This is a size, basis size. And explain example of target structure taken from the ETNF platform, which provided a benchmark set. These targets range from small and long RNA molecule, each with potential application in different biological contexts. For example, small RNA structure, including aptamer, which bind and recognize specific targets, much like antibodies. In construct long RNA sequence, a long RNA structure are involved in variable biological processes. Since this problem was first provided in 1994, advances in computational technologies have gradually expanded the size of RNA structure that can be solved. If we could reproduce the large and more complex secondary structure, we would be able to artificially design RNAs with diverse biological functions. However, there are currently no method that can effectively solve problems larger than about 400 nucleotides. Therefore, designing RNA that perform the diverse biological function still requires further methodological improvement. To solve this problem, we use Ising machine. As you know, Ising machine, including quantum annealing machines, are expected to as efficient solvers for combinatorial optimization problem. To tackle combinatorial optimization problems on the Ising machine, the objective function and constraint must be mapped onto QUBO, quadratic unconstrained binary optimization. However, the objective function of the RNA inverse folding problem is not explicitly defined. Therefore, it is necessary to estimate and formulate the objective function in order to perform optimization using Ising machines. To solve this problem, we focused on the factorization machine with annealing FMA. FMA is a black box optimization method that employs the factorization machine FM as a surrogate model and Ising machine as an optimization solver. This is flow of FM. FM consists of 5 main steps. First, we create a training dataset input and output. Second, training the factorization machine using this training dataset. The FM express the objective function as a quadratic function of binary variables, similar to the formulation used in QUBO. Therefore, Ising machine can sample solution using FM. Next, we evaluate the output of obtained solutions. The dataset obtained from the newly acquired solutions is added the original dataset, original training dataset. Through this iterative process, the FMA can explore solutions efficiency even when the objective function is not analytically defined. Because the FM consists of binary variables, when integer variables or other types of input data, such as graphs and strings, are used, this data should be converted into binary variables before training the FM. In previous study, integer to binary encoding and binary variational autoencoder have been employed for this purpose. So what is a binary integer encoding? There are major binary integer encodings. The one-hot encoding requires L binary variables to express the L integers. The way of expression is a position where the binary variables take 1. Next, the binary encoding expresses the integer in binary form. Thus, the binary encoding needs log base 2 of L. The domain wall encodings has proposed in 2019. The encoding requires L-1 variables. The integer were expressed by the position of the boundary between 1 and 0. The relationship between binary integer encoding and FMA was investigated. In previous study, for problem with real valued variables, this study converted the variables into binary variables using binary integer encoding methods, one-hot, domain wall, and binary encoding, and then applied FMA. In terms of energy error, which defined that difference between the energy calculated by FMA and the true energy, the authors found that the energy error distributions obtained with one-hot encoding was narrow and more concentrated in the low error region than those of the other binary integer encoding methods. In terms of the number of black box function calls, one-hot and domain wall encodings exhibited similar performance, whereas binary encoding shows slow error convergence and low efficiency. The effect of binary integer encoding on discretized array variables has been reported. However, the effect applying binary integer encoding to categorical variables without any inherent order among their value has not been investigated. Because bases of RNA, which are categorical variables without inherent order, must be represented as binary variables. So, the purpose of this study is to investigate the applicability of FMA to the RNA inverse folding problem, and to evaluate the effect of assigning unordered bases to integer variables. For this purpose, we conducted the simulation experiment. The target secondary structure is wrench. On the secondary structure, the sequence consists of 27 nucleotides. This is the secondary structure. And when the secondary structure is written in dot bracket notation, it takes this form. The left and right parentheses represent a basis for that form hydrogen bonds. The objective function value was defined as the sum of two objective functions. One is the maximum free energy, MFE, when the predicted secondary structure is formed. And the other is the distance between the target and predicted secondary structure, d over n, where d is the number of mismatched bases between the two structures, and n is the total number of bases. In this example, there are two mismatches between the target and the predicted structure. Therefore, F2 is 0.074. And lambda is a hyperparameter that takes a positive value. We tested different assignment between bases and integers. There are 24 possible mapping, and applied both one-hot and rainwater encoding method in this study. This is a parameter of FMA. We use the fixed as amplifier as Ising machine, and the coefficients of FMA is normalized by maximum coefficients. And hyperparameter lambda was set 100 and 10,000. We choose three evaluation metrics. First is number of successful solutions. The number of trials out of 5 that produce a solution with a distance of 0 between the target and predicted secondary structure. In other words, F2 equals 0. The second is the average FE, in other words, F1, of successful solutions. The third is the frequency of occurrence of each basis among the successful solutions. Okay. Let us examine the result obtained using the one-hot encoding method with lambda equal 10,000. This is assignment, number of successful solution, and average FME and frequency of each base. According to this table, the successful solution were obtained using FMA. And regardless of the base to integer assignment, the overall distribution of bases was similar. Nevertheless, because lower energy is favored, bases tend to cluster around GC pairs, which contributed more energy stable, like these cases. Because the GC pair shows the lowest free energy among the base pairs, because the GC pair has 3 hydrogen bonds. Next, let's move on the result obtained using the domain water encoding and with lambda equal 10,000. A successful solution were also obtained using FMA, and the bases mapped to 0 and 3 had the highest and second highest frequency when these two bases were capable of forming hydrogen bonds highlighted in this color. For example, in this case, the Z and C are assigned to 0 and 3. The frequency of each base is also high. And in this case, the A and U are assigned to 0 and 3. This pair also high. This is characteristics. So, and the following the frequency of some dynamically stable base pair, the average MFE decreases accordingly because GC pair is frequency of GC pair is high. The free energy becomes low, like this pair and this pattern. So finally, this is a result of binary integer encoding method is domain water encoding and with lambda equal 100. According to this result, because the hyperparameter lambda is small, in this pattern, the overall MFE values become lower compared to the case when lambda equals 10,000. Because the decrease in MFE has strong influence on the objective function value, the frequency of GC pair tend to increase like this. This is a conclusion and future work. FMA successfully generated a feasible RNA sequence that fall into the target secondary structure. When the influence of MFE in the objective function was small, using domain water encoding result in the certain bases being generated depending on the assignment between bases and integers. It was shown that results varied depends on the types of binary integer encoding used. It was suggested that this may be due to the ordered transition between integer values. We further investigated the underlying mechanisms of this phenomena and extend the approach to other target secondary structure. Thank you for listening.
Okay, time for questions.
Hi. I had one question about the application of this this technique of the inverse RNA folding. So it's like a technique for enzyme design or drug design, or what do you use this for? And second part of the question, basically, does this approach then, if that's the application, extend to also giving like protein secondary structures and determining those, or is that much more complicated?
So you mean these methods like FMA can extend the other program? Yes. If you didn't make a formulation, you can use the FMA.
Also for proteins. Okay.
Okay.
More questions?
Mine's kind of a follow-up. So you know how in this case you had nucleotides which interact with each other via hydrogen bonds. If you have the set of amino acids, there are more types of bonds that you can put in. I have a feeling your answer will be yes, but is that easily encodable? Can you put different flavors of bonds rather than just hydrogen, like disulfide bridges, polar bonds, etc., van der Waals? That's my question.
In this case, the most frequency and the most important bond is hydrogen bond. There's some pattern, some chemical bonds also appear, but it is very rare in RNA secondary structure. So it is okay to ignore these bonds.
But if you were to adapt it to the protein folding problem,
okay,
where those bonds become significant,
okay,
could you do that easily using this method?
If you use a protein folding problem, FMA can use, because the FMA didn't recognize some types of bonds. FMA just recognize input and output data. Okay, the objective function recognize these some types of bonds.
Okay, cool, thanks.
Thank you. I'm just not familiar with this technique FMA. Could you explain it a little bit more in depth? Especially, I didn't understand how you reuse the results into the input data.
So you want to know the reuse of dataset?
Yeah, just if you could tell me what is FMA in the first place. I'm not familiar to it. Like in the slide before, the one, yes, exactly here. I just don't get why you use the shots you measure back into the training data. Did I understand correctly?
Yeah, we add new acquired dataset in the original training dataset. So number of dataset increase through this iteration.
Okay. Okay, I will ask you later maybe. Thank you.
Thank you.
Any more questions? Okay, then we can thank again the speaker.
Thank you very much.